Hello everyone, hope you all are doing well. So today we are going to do topic 66 C. Topic 66 C of paper 2, where we are discussing major pests and diseases and their management. So first of all, the disease we are going to discuss today is mango malformation, and we have discussed some disease in topic A and B, so you must see it. And today we are going to do third part of it, that is topic 66C. So first of all, we are going to discuss mango malformation. So it is a fungal disease, you should know this. It is a fungal disease, first reported from Bihar. It was first reported in India from Bihar. Most common in northwest India. It is most common in northwest India. Now what are its types? So it is mainly of two types, that is vegetative malformation and floral malformation. So first of all, we will discuss vegetative malformation. So more commonly found on young seedlings. It is more commonly found on young seedlings. These seedlings produce small shootless bearing these seedlings produce small shootless bearing small scaly leaves with bunch type like experience in sweet apex, with bunch type appearance, and this can be seen, bunch type appearance can be seen in shoot apex. Seedlings remain stunted and die, while those getting infected later resume in for normal growth. What happens? Seedling remain stunted. The seedling remain stunted and they normally die, while those getting infected later resume normal growth. Which infected later, not in earlier stage but infected in later stage, can resume normal growth. So this was about vegetative malformation. It is on young seedlings. Now moving to second, that is floral malformations. Floral malformations. These are the malformation of panicles. Severity of malformation may vary on some shoot from light to medium or heavy malformation of panicles. So severity varies from light malformation of panicles, light to heavy malformation. Heavily malformed panicles are compact and overcrowded due to larger flowers, and these heavily malformated panicles are compact and overcrowded due to larger flowers. They have larger flower, that's why they are compact and overcrowded. They continue to grow and remain as black masses of dry tissues. They continue to grow and remain black masses of dry tissues. It seems to appear as black masses of dry tissues during summer, and this is especially during summer due to dryness. But some of them continue to grow till next season. But some continue to grow to next season. So this was what? Floral malformation. Now what are its management? How can we manage malformation? Let me rub it only. Management of malformation. Disease plants should be destroyed. First of all, destroy disease plants. Okay, and we should use disease-free planting material. Use of disease-free planting material. Secondly, cyanistics from infected trees should not be used. Cyanistics which are infected must not be used. Fit cyanistics should be used. As soon as disease appears, the affected terminals along with basal healthy portions should be removed. As soon as disease appears, the affected terminals with basal 15 to 20 centimeter of healthy portion. Suppose this is affected terminal, so we will also take 15 to 20 centimeter of healthy portions around affected terminal and we will remove it. Or you can simply say you can prune it. If more than 25% affected plant, deblossoming at bud stage should be done at delay the flowering. If the more than 25% affected plants, the plant is affected by more than 25%, then deblossoming at bud burst stage should be done to delay the flowering. This should be done to deblossoming to delay the flowering. We can spray Planofix. At 200 ppm during the first week of October. This we need to do first week of October, followed by deblossoming at bud burst stage recommended, and after then we will deblossom. After spraying Planofix, we will use deblossoming. A single foliar application of 100 ppm cobalt sulfate. We can also spray 100 ppm cobalt sulfate prior to flower bud differentiation. This is prior to flower bud differentiation, and this can be done to reduce floral malformation. This can be done to reduce floral malformation, that is on panicles. So this is the management. This is what you can do, and a single foliar application of 100 ppm is more than enough to combat floral malformation. So this was about mango malformation. I hope you understand this. Now moving on to the next topic of this session, that is black sigatoka of banana. Black sigatoka of, it is caused by fungus, that is Ascomycete fungus. Okay, what are the symptoms? Red or brown black streaks. We will see red, brown, or red, brown, or black streaks appear underneath of younger leaves. This will appear at underneath of younger leaves. Lips show elliptical spots. Lips will show elliptical. With center of these spots turn grey to black, and the center of these spots will turn grey to black. Also can see yellowing of leaves, and also we will see leaf yellowing. Leaf will become yellow in color. Yellowing of leaves is common. Spots often coalesce to form large irregular patches of dried tissue. These spots often coalesce. Coalesce means they will merge together and form large irregular patches of dried tissues. Suppose this is a banana leaf. Okay, first of all you will see a normal black spot in the center of elliptical structure, and second, suppose this is here, this will start coalescing and this will become a dry mass of dried tissue. Rapid drying and deflation are the characteristics of feature of this disease, and the main characteristics feature of this disease is rapid drying and defoliation. Are the main characteristics of this disease. So this was about symptoms. Now how will you manage this disease? It is black sigatoka of banana. Management. First of all, remove and destroy affected parts or affected leaves. Remove or destroy affected leaves. Secondly, keep banana field weed free and remove suckers timely. Banana field we should be weed free, and here we should remove suckers. Suckers should be removed at timely. Avoid planting at close spacing. There should be no close spacing of banana plants. It will lead to more rapid growth of disease from one plant to another. They will contaminate other plants also. Proper drainage and avoid waterlogging, which favors infection. There should be proper drainage in the field, and we should avoid waterlogging. Avoid waterlogging because waterlogging leads to growth of fungus. It leads to growth of infection. We can use mango jab, copper hydroxide, and banana misting. Oil. We can also thiazoles, and these can be used for chemical management. These chemicals are used for removal of for management of black sigatoka. Some resistant varieties can be also used. Resistant varieties such as FHIA. These are some international resistant varieties, 01 and FHIA 02. 01 and FHIA 02. So these are resistant varieties which can be used. So I hope you all understand this management and symptoms. Now moving on to next disease, that is powdery mildew of roach. Powdery mildew of roach. It is caused by Spearothosia penosa. Its symptoms are: the symptoms appears as greyish white powdery substance on surfaces of young leaves. You will see a greyish white substance such as powdery substance at young leaves. Powdery substance at young leaves, shoots, buds, etc. Greyish white powdery substance. This the name here. It gets its name from this symptom, that is powdery mildew. It appears as powder Infected leaves may be distorted and some leaf drop may occur. Infected leaves may be distorted, or you can also see leaf drop. Flower buds may fail to open, and those do may produce poor quality of flowers. First of all, flower bud do not open, and those which open produce poor quality of flowers, poor flowers. It occurs almost any time during the growing season with temperature and mild. Any time with mild temperature favors it. And relative humidity is high. Mild temperature with high relative humidity favors this disease. And it can be almost occur any time during the growing season. It is most severe in shady areas and during cooler periods. It is most severe in shady areas where shade is there with cooler periods. When there is cooler period and shady areas, it enhances infection. Now, how will you manage it? Management of powdery mildew of roach. Management: collection and burning of fallen leaves. Collect and burn fallen leaves because it can be spread, help in spreading infection. Spray with wettable sulfur. Wettable sulfur 0.3% or carbendazim at 0.1%, and we can do 2 to 3 sprays of both around interval of 15 days interval. And we could also use sulfur dust. Sulfur dust at 25 kg per hectare. Use of sulfur at higher temperature conditions will be phytotoxic, but we should not use sulfur at higher temperature because it can become phytotoxic. So we must keep in mind this. And these are the management of powdery mildew of roach. Hope you understand all this. So that is all for today. For any queries, do comment. If you like it, please press the like button. Do share and subscribe. Keep making notes. That's all for today. Have a nice day. Thank you.